Okay, let's just get started. Today we're gonna talk about a new chapter, and it's the most difficult chapter in the whole course, I think. But we're gonna start slow. So we're gonna start with metabolism, and let's see the next one. So in the beginning of the semester, the first class, remember we talk about how to qualify as a living being, you have to have energy, right? And the ability to acquire and use energy. So hopefully at the end of this chapter, you guys will be able to answer the question in green, which is eating and breathing connected? In another word, um, um, why do you need oxygen? So this is a very critical question. A lot of the students that I had, they will say, oh, because we need to oxygenate our blood, and our muscles need oxygen and stuff like this. But we need to get down to the cellular level. Why do we need oxygen? So at the end of this chapter, hopefully you'll be able to answer this question really well. And the truth is, eating and breathing is connected. Okay, if you don't breathe or you don't get oxygen, the food you eat is useless. You cannot break them down into energy for your cells to use. Okay, so just a quick definition: energy is the capacity to do work or supply heat. Okay, and there's two kinds of energy. One is potential, the other one is kinetic. So kinetic, you should know already, is the energy of movement. Okay, and potential energy is stored energy. It has a lot to do with location. Okay, for example, the one I have is electrons in the valence shell has the most potential energy than any of the electrons in the inner shell. Okay, because the valence shell's electron not attracted to the proton in the center of the atoms that much, so they can participate into chemical reaction, and they actually have more potential to participate in chemical bonds. Okay, so the chemical bonds store a lot of potential energy, and this kind of potential energy is called chemical energy. Now, when we talk about, I'm missing a few slides. Give me a minute. Why is it not? It's showing up in a minute. Okay, so when we talk about energy, we're dealing with metabolism. Okay, metabolism is all the chemical reaction that happen inside of you. These reactions can divide into two categories. One is catabolism or catabolic reaction. The other one is anabolic reaction. Okay, and all of these reactions are catalyzed by enzymes. Okay, just remember all the metabolism reaction, catabolic or anabolic, catalyzed by enzymes. Now we come into a new term for you guys. It's called free energy or Gibbs free energy, and the symbol for it is G. So free energy is the amount of energy that's available to do work, and the way they calculate it is they look at how much energy is in a chemical bond, okay, the total energy in the chemical bond, and then they subtract the heat that will be lost when you break that bond, because the heat that's lost is not able to use for work. Okay, so then the Gibbs free energy is calculating the available energy to do work that way. So here's a quick equation. Now when you see a little triangle, it just means change, delta. So the little triangle is called delta, and it's just telling you the changes in free energy. So the equation in red showing you the changes in free energy is this enthalpy and then temperature and then entropy. So you don't have to worry about this equation. But in general, what you need to know is that the change in free energy will be negative if the chemical reaction is exergonic and spontaneous. Now, exergonic and spontaneous should be remembered together. So any exergonic reaction will be spontaneous. Okay, I'm going to talk more about this in a minute. And on the other hand, if delta G is more than 0, that means it's positive. Now in this case it's negative. This case is positive. Then the energy is, then the reaction is endergonic. That means energy has to be invested for this reaction to happen. Now the way to remember is ender means enter, right? Energy enter. Exer is exit. So energy is leaving the system. So this is the way to remember it. Now when we talk about catabolic reaction, catabolic is breaking down polymer, right, into monomers. So hydrolysis reaction is used to break down these kind of macromolecule. Remember we talked about that? And the way hydrolysis work is by taking a water molecule to split the H and OH, and you put it here, and that way you break down the polymer. And when you break a bond like this one, when you break any chemical bonds, It's always energy is released, remember, because these chemical bonds store a lot of potential energy called chemical energy. So when you break them, some of them release as heat you can't use, some of them release as energy you can use. So this is an exergonic reaction. Okay, so catabolic reaction is always a exergonic reaction. So delta G is always less than 0. So here's a picture of catabolic reaction. So what you're looking at is in the beginning, the molecule that you have, the energy level is at right here. After the bond's being broken, all these energy is released, right? Remember the chemical energy in the chemical bond is released. So the resulting product has less energy than what you start with because you're breaking all these bonds and releasing all these energy. So delta G in this case will be negative. And exergonic is always spontaneous. So you're releasing energy. So the way I remember this is exergonic reaction is releasing energy. If you think about energy as money, so spending money is very spontaneous. You don't need to plan, right? It's always good. Making money, on the other hand, is not the same thing. So anabolic reaction is the reverse of catabolic, where you're using dehydration synthesis to put monomers together, right? So you're excluding a molecule of water and you join two monomers together. And to form a chemical bond like this, see, to form a covalent bond like this, it always requires energy input. So you have to invest energy into this bond. Then it becomes chemical energy. And so for anabolic reaction is always endergonic reaction. And if you look at the curve, so if this are the monomers that you start with, after forming all these chemical bonds, you're investing a lot of energy into these chemical bonds. So the end product, which is the polymer, will have more energy than the monomers that you start with. Okay, so delta G here will be greater than 0 because you look at this, the difference between here to here is a positive difference. Okay, and if the analogy applies, this is like making money or invest or depositing money into your bank account will go positive, right? But catabolic reaction is like spending money, so your bank account is going to go negative. That's how you're going to remember. So in anabolic, delta G is positive because you're depositing money into your bank account, so your account balance will be positive. Okay, so remember that. Now, if you look at this type of reaction, now remember the way to look at it, are you breaking down big polymers into monomers, or are you making polymers from monomers? That's how you can tell whether it's endergonic or exergonic reactions. So if you look at this, okay, are you building up or are you breaking down? So what kind of reaction is this? Is it endergonic or exergonic? So since there are only 4 of you, so it should be pretty straightforward. Yep, this is exergonic because you're breaking down this glucose into individual CO2, okay? So endergonic, exergonic, or anabolic, catabolic reaction, they always occur at the same time. That means they always couple. So the energy that is released by, let's see if I have it here. The energy that is released, let's do it again, by the catabolic reaction, remember, catabolic always, when they break chemical bonds, they release energy, is always used by anabolic reaction to build stuff. So the energy that is released from catabolic reaction is used directly by anabolic reaction to build stuff, right? So that's what we call energy coupling. Now, when you talk about all these metabolic reaction, all these metabolic, anabolic reaction, catabolic reaction, whatever it is, they are catalyzed by enzymes, like what I told you guys earlier, right? So, and not just that, the enzymes that for a certain reaction always arrange in what we call biochemical pathways. Look at this. So enzyme number one will convert this substrate, the initial substrate, into a product, which is a triangle here. And this product from enzyme number one will become the substrate for enzyme number two, and so on and so forth. Can you see that? So it's kind of like an assembly line that they just work on it sequentially like this. So when they organize the enzymes like this, it's way faster when they want a reaction to take place, right? So this is called biochemical pathway. The substrate or the product from one enzyme become the substrate for the next one, and they all line up like this. Now, so we're gonna talk about cellular respiration, focus on that, and the purpose of cellular respiration is to break down food to harvest its energy, and so in the form of ATP, so then cells can use this ATP for whatever um, task that they're trying to do. Now, if you look at the equation for cellular respiration, that's what I have. You start with some sort of food, but in this chapter, we're going to focus on glucose because it's easier to look at, okay? So you start with glucose, and you mix with some sort of certain amount of oxygen. So what you will do when you combine these two together, you're going to go through cellular respiration and release some of the ATP, right? Some as ATP, you break all these chemical bonds and you release some as heat. And then the end product is you're going to release CO2 and H2O. So the equation for cellular respiration is sugar plus oxygen, and then you have an arrow. I think I have that later on here. I have an arrow and then show that, then it turn it into CO2 and H2O. Now, when you look at this equation, you go, it's really abstract, but think about it. When you exercise, when you start running, okay, what do you guys experience the first 5 minutes or 10 minutes after you start running? Can somebody tell me? Come on, what are some other stuff besides you get tired? Well, the first 5, 10 minutes shouldn't be tired already. Who is that say tired? Oh, right? If you've been exercising for an hour or 30 minutes, then you start getting tired. But the first 5 to 10 minutes, what you start experiencing. Well, an area is right on it, hot and sweaty, right? Okay, so let's look at this. When you break down glucose, when you exercise, right? When you exercise, you need more energy. So then well, the one thing you didn't say is you're going to breathe a lot harder, right? Your breathing rate is going to pick up. So it's because when you start exercising, your muscle need more energy. So your body will start breaking down glucose more. So when they do that, they get more energy for the muscles to use. But at the same time, if you are metabolizing or breaking down more glucose, you need more oxygen to go with it, right? So that's why you start breathing really hard because you're going to need more oxygen to burn these glucose and turn it into ATP, okay? And um, and of course, when you breathe really hard, you also breathe out more CO2. So you get rid of this part here. Now your sweaty part is this here because you get more water too. So you're getting sweaty, you're breathing harder, and what is the other thing? Hot. Why are you getting hot? Because remember I said that when you break these chemical bonds in glucose, besides releasing ATP, some of the energy in the chemical bond is released as heat that you can't use. So that's why when you do cellular respiration in here, during your exercise, when you increase the rate of cellular respiration, you breathe harder because you need more oxygen to metabolize these glucose. And when the ATP is released for the muscle to use, always come with some heat because of the chemical bonds that's broken, right? And then you breathe out more CO2 and then you start sweating, which is the water. So it all adds up. Okay, so this is a quick, since we're not going to do photosynthesis, the last chapter. So this chapter that we're working on right now will be our last chapter for the semester, okay? And but for photosynthesis is the reverse of cellular respiration. Remember, in cellular respiration, you're breaking down glucose to release energy. In photosynthesis, the plants are using solar energy to build sugar. Okay, and so here is glucose. If you remember, we have C6H12O6, remember? So in photosynthesis, all these energy from the sun is converted by the plants in these carbon-carbon bonds and carbon-hydrogen bonds. But anyways, all these chemical bonds. Um, so the overall equation we talked about already for cellular respiration. Now, from now on, cellular respiration um, is same as CR because it's too long to type. Okay. So for CR to happen, you have to break down glucose. You're going to trap the glucose, trap the energy in the form of electrons. So, and then you use this energy to make ATP. So the whole goal of cellular respiration is to take the electrons from food. Okay. A long time ago in the beginning of the semester, chapter 2, I believe, when I talk about electrons are the hot stuff in the universe, it's because electrons, imagine them flying around, right? And you can imagine they have a lot of energy. So human, when we eat food, what we're trying to do is we try to steal electrons from food that we eat. All those flying electrons that you can imagine in your head is what we are after when we eat food. Now, enzymes is the one that makes everything possible, right? Um, and cellular respiration is no exception. So when we use enzyme to go through many steps in cellular respiration, the advantage is that the energy will be released in very small, manageable packets. Okay, and so it can be captured by ATP. Some of them, most of them, will be captured as ATP, but like I said, the rest will be released as heat. That's why when you start exercising, when you start breaking down those glucose more, you start heating up and getting hot. And why is this good? Enzymes plus many steps in cellular respiration. Since every time you break down a chemical bonds, you release heat. If you do if you release all these energy too quickly, you overheat. Okay, and plus, if you release it in a very small package, then you can capture most of the energy. Now, here's the first major concept you need to understand. In metabolism, what we are doing when we have anabolic and catabolic reaction, we are transforming energies and matter, okay? And all these transformation is subjected to the law of thermodynamics. Now, thermodynamic is a huge word, but basically what it's talking about is the energy transformation that we see every day in our life. And we're just going to talk about two laws. So the first law of thermodynamics is about the law of conservation of energy. It talks about energy cannot be created or destroyed, but it changes from form to form. Okay, so the total amount of energy in the universe is constant. So let me give you some example. So when you rub your hand, when you like right now it's really cold, right? When you get cold, your hands get cold, and you rub your hands. This is mechanical energy when you're rubbing your hand, right? Mechanical energy that will turn into thermal energy. You turn it into heat, right? So let's look at some of these. So when plants use the sun energy, which is the solar energy, and make it into a plant like this, grow leaves or do whatever, they turn it into chemical bonds. The energy become chemical bonds, so it become chemical energy. So you transform solar energy into chemical energy in plants. Okay, and when you cook at home, you're using electric stove or whatever it is. Let's say it's electric stove. You're turning electrical energy into thermal energy. You're heating up stuff, right? Here, same thing. A person eating an ice cream and then go out for a bike ride. The energy in the ice cream is chemical energy in food, remember? And then riding his bike is a mechanical energy that is physically moving. And the last example here is the gasoline you use for your car. They are chemical, right? So they have chemical energy. So then after putting in your car, it becomes mechanical energy because the car can move now. Can you see how all these examples follow the first law of thermodynamics, where energy just transform into different forms and it's never lost? So it's conserved in that way. Now, during cellular respiration, the energy from food, which is chemical energy, right, in the chemical bonds, is converted into chemical energy again, but it's not in food anymore, it's in ATP. So in this case, even the form of energy is the same, but the matter is different. It was food and then become ATP, okay? So like I said, the heat here is released every time you break a bond. Besides ATP, you also have some heat left. And like I said, heat cannot use to do work, but it's mainly used to warm us up. Okay, so most of you vote for B. Good. So remember that. Now, the second law of thermodynamics talks about entropy. Now, if you haven't seen the word entropy before, it's just talking about the disorder, the disorderness of the universe. So they're saying that this universe actually, energy transformation we talked about in the first law, always proceed spontaneously to make matters less order. So in another word here, this is order. So the example is this is very nicely organized, right? So the energy transformation always go toward a chaotic form of matters. So in cellular respiration, remember glucose is very structured. You have this carbon all linked together with some OH and H. When you break down glucose during cellular respiration, you convert that structure molecule into CO2 and H2O, which are less order. So because they're gas, now remember glucose is solid, so they're less messy in a sense. But H2O and CO2, they're mostly gas, so they can fly around and it's more messy. So what it's trying to tell you, the second law of thermodynamics, is that reaction always go toward messy. So the messier, the better. So here's another example of second law of thermodynamics. As you see, when this little guy, this cheetah, after he ate a piece of meat, okay, now remember that meat is muscle fibers, you have all these stuff, very structured. So he broke it down in his stomach and then it becomes energy, right? So the piece of meat turned into heat, turned into CO2, H2O, which is way less orderly than the meat that he ate that he started all this with. Okay, so this is telling you that cellular respiration. One thing, number one, cellular respiration um, follow the law, the first law of thermodynamics, because it transforms energy from food into ATP. Number one. Number two, it also follow the second law of thermodynamics because um, it is spontaneous in the sense that the reaction goes spontaneous because the glucose that it used to start with become more messy. So that's why it followed the second law of thermodynamics. So cellular respiration is spontaneous because you're going into the same direction. It's the second law of thermodynamics. Now, before we go any further, is there any question? Before we talk about cellular respiration, we have to know a few things. So this is what we're trying to cover. We're not even going to talk about cellular respiration yet today. We're going to talk about redox reaction. We're going to talk about what are electron carriers. We're going to talk about the structure and function of ATP. Okay. Now, redox reaction is a term, means reduction. RED is reduction reaction and oxidation reaction. The reason it's called redox or redox, whatever you want to call them, is because these two reactions always occur at the same time. So the way to remember is this. Pen. I think in the textbook or somewhere it says this. So to remember this is remember oil, reg, and that's what I've been remembering all these years. So oxidation is the loss of the electron. Okay, see here? Loss of electron is oxidation. So oil is oxidation, is the loss of electron. Reg is reduction, is gain of electron. You have to find a way to remember it because otherwise you would think, oh, reduction means reduce, you're losing electron. No, actually it's the other way around. And they always occur together because the electron will not just leave one molecule randomly, right? It will only leave a molecule because it's pulled away from the other. So in the beginning of the semester, we talk about sodium chloride, remember? Chloride has such a strong electronegativity than sodium. So the electron from sodium get pulled or get stolen by chloride. So then at the end, you get two ions. Remember that? You get a cation here, which is positive because this guy lost his electron. And this become anion. I hope you remember some of that. Right? So in this case, you see how the electron don't just randomly leave sodium unless it's pulled away from chloride, right? So this here is an example of redox reaction. Now, since electrons contain energy, right? Remember the picture you should have in your head is that the electron is flying around the proton, right? So that energy, so when a molecule gain an electron, besides that electron it also gain the energy that is associated with that electron. Okay, so it's kind of like if you marry somebody rich, you're marrying that person with the money that comes along with it. Okay, so it's the same thing. So a reduced molecule gain electron, right? Gain electron, also gain the energy that goes with it. But the other thing is how you can't see electron, right? So how do you keep track? How do you know whether electron move or not, right? So in a molecule sense, what you're going to look at is a proton because electrons, just like a minor, they don't travel alone. They always travel with hydrogen, okay? So to keep track of electron movement, you have to keep track of the hydrogen. Okay, so same thing, oxidized molecule, you will lose an electron, which is same as a proton because that's what you keep track, and you have less potential energy. So the summary is the reduced form of a molecule always have more energy. Now here's an example that you need to work on. So let's look at this question and see if you know what it's talking about. Okay, so that's a very good question, Anne. So let's finish this question and I'm going to talk about the question you have. Okay, let's see. A, you said, oh, most of you vote for A. You said A is being oxidized because you see this H. And so in here is the answer for your question. The proton that it refers to in the last slide is not actually a proton. Remember I said that if you look at hydrogen ion, if you look at your periodic table, hydrogen only has, if you look at this, hydrogen only has one electron. So when you look at H plus, what you're saying is this is poor little hydrogen lost its only electron. Okay, so there's no electron. So when it doesn't have an electron, it's H plus like this. So a lot of times we refer to H plus, which is a hydrogen element without the electron, a proton, because there's only one proton in it, right? Remember this? So there's electron here, and there's one p in here in the center, right? So since the electron is gone, we just call it a proton because it's what is left. And hydrogen doesn't have any neutron. So when what we refer to in the last slide, the proton is talking about the H plus. And H plus doesn't have electron. That's why you can travel with the electron. Okay, so you guys got it right. A is correct. So you see that A loses its electron in the form of hydrogen ion, and then it gives this hydrogen to B. So B is being reduced because it gained. A is oxidized. Now let's look at a real life example. So Anne, I see a question. So let's finish this question and I'm going to try to answer the question that you have. So this one is like I said, it's a real life example. So you're looking at this molecule here, malate, right? And you can tell at the end of the reaction, the NaH gain an electron, right? So you know that NaH, so this has to be correct, is reduced because it gained an electron. Now L-malate is oxidized. Is that correct? So since this guy gained an electron, this molecule has to lose one in the form of proton. If you check, so it's this one here, right? That is being moved to this guy here. So then B is also correct. And then you say L-malate has more free energy than oxaloacetate, which is also true. Why? Because this one has the electron. This one is missing the electron, right? Remember the electron is the one that carries the energy. Now then I say D, so then the answer is E. All of the above is correct. So let's answer In's question. Now In, I think the answer is this. When we talk about electrons in oxidation reaction traveling through protons, hydrogen, we are talking about big molecules like this, see, like malate. When we talk about the example of sodium, and chloride, you can already guess what I'm going to say. These are simple elements, right? So if you put sodium elements next to chloride close enough, the electron, I don't know for sure, I think it's a physics question, can just go to the electron shells on chloride. It doesn't need hydrogen or proton to travel with it. But when you're dealing with an organic molecules in cellular respiration like this, you will have to have that. And that's why we're going to talk about in a minute. NAD is actually a designated electron carrier. So electron in biological system cannot travel freely because you can imagine in the body it'll be kind of crazy, right? You have all these stuff going on. If it's not traveling with something in a specific way, it can get lost, right? But in the sense of sodium and chloride, it's a simple chemical reaction, not in a biological system. Um, so it's more chemistry in a test tube or physics, whatever, that is just naturally happen. So here is the energy that we've been talking about that we're trying to steal electrons from are in the chemical bonds in glucose. In particularly, we're looking at the carbon-carbon bond and the carbon-hydrogen bond. And the reason being is because if you remember, carbon and hydrogen has very low En, okay? Very low En. So just like stealing candies from babies, right? It's easier to steal electrons from these combination than, let's say, if you try to steal electrons from oxygen, which is hard because oxygen has really high En. Can you see that? So in the sense that these elements has low En, low electronegativity, so they hold their electrons very loosely. So it can be stolen by us. That's the whole thing. So that's why we can only eat carbon-based food molecule. Water can never be enough for us as food source because there's no carbon-carbon bond, and we are not strong enough to steal the electrons from oxygen in water. So here is another concept. When electron move from a toward a more the stronger En atom. For example, the electrons from this carbon-carbon bond, if our body steals it and put it in with oxygen, so it's moving from a low En to a high En element, the oxygen, since it's holding the electron so much stronger, the energy is going to be released because the electron moving from here to here will lose its potential and energy because it can't fly around as freely as before because oxygen has such high En. So chemical energy is released in a redox reaction. So let's look at this again. When you move, so in a sense, NAD here is more electronegative than L Maule. That's why it will give up its electron to NAD. Okay, so when electron is moving from L Maule, which is less electronegative, to a more electronegative NAD, some of the energy from the electron will be gone because it's being held so much tighter. So the part of the potential energy will be gone. Okay, so that energy can be used to do work also. So here's the summary, the last sentence. So chemical energy is released during redox reaction, right? You're moving electron from an element that has lower En to an element that has higher En. So here's what it is. So glucose showing you has the lowest of all En, and then eventually the electrons in glucose will travel through these steps, reaching oxygen at the end. And so oxygen has the highest En of all. So then potential energy of the electrons in glucose gradually be lost through the transfer of electron during the redox reaction. Now, this is the equation for cellular respiration, right? You're gonna mix some glucose with oxygen, and then you have water, CO2, and of course you have ATP. But one thing I didn't write here is what I'm missing? Heat, right? So we can write heat here also. Now, if you look at this reaction here, it is a redox reaction. Can you tell me what is being reduced and what is being oxidized? Okay, so Harley got it right, and then you guys also got it right. So uh, let's look at this, okay, equation here. So if you look at glucose here, which is this guy, right? Look at all the oxygen. Look at all the hydrogen here. So glucose end up becomes CO2. So all the hydrogen here will go to oxygen, and that will become H2O. Can you see that? So this is the O from oxygen, and then all the H is from this. And then when this is all gone to oxygen, it end up with CO2. Good. So glucose is being oxidized. Remember, oxidized means losing electron, losing its energy. So the goal of cellular respiration is to get all the energy from glucose, to steal all the electrons from it. And oxygen is the one being reduced, gaining all the hydrogen. And why is oxygen? Because I told you already, oxygen has really high En. So compared to glucose, right? So then this hydrogen will always go there because it has really high En. So breakdown of where is it? So glucose or cellular respiration is a redox reaction. You oxidize this, releasing its energy, and then you reduce this. So when we release energy from glucose, what we're doing is we're breaking all the chemical bonds. In particular, we're going to keep track of the carbon-carbon bonds. Okay. We're going to look at carbon-carbon bond. This one. So now here's a key thing to remember. When you break a chemical bond, like a carbon-carbon bond, single bond, two things happen. Number one, remember between these carbon-carbon bond, you have two electrons, it's a single covalent bond. So they're sharing electrons, right? So these electrons, when you break this bond, it's not going to be shared by these two elements anymore. So you will have to have an electron carrier to carry the electrons after you break the carbon-carbon bond. When you break the carbon, so you have the electron, you need to be carried by NAD plus. And plus, when you break the carbon-carbon bond, the energy, the chemical energy in the bond is going to be used to make ATP. So two things, remember, every time when you break a carbon-carbon bond. Now, so what we're trying to do here is, remember glucose is 6 carbon. So we always represent it like this. Just simplify it so you can see it well. 4, 5, 6. So in the process of cellular respiration, you're going to see how, with all the steps that are involved, we're going to break each one of these bonds and release all the chemical energy in the form of reduced electron carrier or ATP. Okay, so just remember this. Most of the time when you break a chemical bond, these two things will happen. Now let's talk about electron carrier, NAD. Now NAD, here, NAD is a coenzyme. Now do you guys remember coenzyme at all? So it's a carbon-based molecule, right? And it's actually part of the vitamin B family. Do you remember? So one of the vitamin B is used actually as an electron carrier. So when it carries electron, so NAD plus is reduced to NADH when it picks up 2 electron and 1 proton. Okay, so that's why you only see 1 H here, but it's actually carrying 2 electrons. So this is the energy carrier. Now, so when I say that is electron carrier, remember electron has energy, so it's also called an energy carrier. So when you go from NAD plus to NADH, we call this a reduced, right? Again, electron, reduced electron carrier. Okay, most of you say A, so you said that good. So A is correct. So the answer is A. Now let's talk about ATP. Well, let's take a break for 5 minutes and then we come back and talk about ATP, okay? 5 minutes break. Okay, let's get back together. Before I talk about ATP, I want to talk about the exam. Now, once again, let's see what exam this is. It's going to be exam 5. Okay, so exam 5 will cover DNA replication. Remember the leading and the lagging strand and the mutation, and also will cover meiosis and genetics. So I will have the exam up and ready by the end of today. So you should be able to take it tomorrow if you want to, and I'll have a week. I'll give you guys a week to complete the exam. Same thing like last exam. You have to. It's a time exam. That means you have to get the exam done in 110 minutes, which is a class period, and it's half multiple choice, half essay. Maybe this time a little bit less essay. Okay. And this will be the last exam, and then after this, um, the chapter that we are covering right now, cellular respiration, will be in the final exam with the other chapters. So this is the last exam, and then we'll go to finals, which is not until May 11th. So you have a lot of time. But next time, which is Wednesday, we're gonna talk about the biofuels project, and this will be the last lab for the semester. Um, So next time it's going to be biofuels, and then on Friday we'll continue on cellular respiration, and then on next Monday there's no class because it's supposed to be exam 5. So I'll give you guys that time to catch up on your work or to ask questions. I will still have study session going on that day. Um, and then on the 22nd we will finish cellular respiration. Okay, so I'll keep reminding you about what we're going to do every time, so you don't have to worry about it. Is there any question about exam 5? If there's no question, let's talk about ATP. ATP is the energy currency of our body, actually of all biological system. And if you look at the structure of ATP, which I have up here, does that remind you of something that you already learned? Look at ATP. What does that remind you of? Yeah, right on. People, good, good. RNA, DNA, right? The only difference is ATP, A stand for adenosine, what is it? Do I have it? Adenosine triphosphate. So it's only have adenine instead of A, in DNA you have the choices of A, C, G, T. But the sugar is the same as RNA, so good, right on. But instead of one phosphate, ATP, since it's triphosphate, the T stand for tri, it has 3 phosphate. Okay, remember a regular nucleotide only has 1, right? And the energy is stored in the phosphate bonds here. in the last one here. And it's because, remember phosphate, all these oxygen is negative. So if you're putting all these negative charges together, what you're doing is like this. You're like compressing a spring because all these negative charge, they want to repel each other. So if it's like you're holding a spring one, you open, you're holding the spring closed. And so ATP, those three phosphate together is very, very unstable. So the bond, if you have hydrolysis going on with water mixed with ATP with some enzymes help, you break the last phosphate off. You see that? And this last phosphate becomes this inorganic phosphate. And just by breaking that last bond right here, you get 7.3 kilocalories per mole of ATP. So that's quite a bit. Adenosine, I don't know. And you should look into it. Tell me why it's called adenosine triphosphate instead of adenine triphosphate. And then let me know, okay? So how much ATP does a cell use? Actually, a cell uses over 10 million ATP per second. So you can imagine, number one, how active your cells are every day, every minute, every second. That's why it needs that much ATP, right? And number two, the cell has to have the ability to constantly make ATP. If not, it can't survive. So the way to make ATP, so if you look at this is what we saw before. When you break down ATP, the last phosphate, so instead of triphosphate, you become diphosphate from 3 to 2. And when you go through from ATP to ADP, you use a special enzyme called ATPase, which is the enzymes to break down ATP. But once it's broken down into ADP, immediately it's get remade into ATP, and then the remaking process of ATP is called ATP synthesis, right? Making ATP. So the reverse is done by this other enzyme. So ATP is constantly broken down into ADP, but then it's constantly being remade back into ATP, okay? Yep, C is correct. So the cycle is constantly going on. Just like what you guys do right now, you spend money and then you make money, so you have to have that cycle going on. Otherwise, it won't work. Now, remember I talked to you about how catabolic reaction always couple with endergonic reaction, right? And the energy released by catabolic reaction we use in the endergonic reaction to make whatever is necessary. But think about this. Your body does not, well, how do I put it? Exergonic or catabolic reaction, the rate of catabolic reaction and the rate of anabolic reaction sometimes is not equal. At a certain time, your body need to break down more stuff than to make more stuff, right? Does that make sense? So the rate of catabolic reaction and anabolic reaction is not equal in your body. So what happens if, okay, if you have more catabolic reaction going on, that means you're releasing a ton of energy, but you have no anabolic reaction going on. You're not building any new stuff. Let's say it's like us, we're getting old, right? We're just constantly burning energy, but we're not building any new muscles or growing taller and anything like that. So what do you do with those energy that's released in catabolic reaction? So the body has a really good and smart way to take care of this. So look at this. So if you have catabolic reaction here, releasing all this energy, right? This arrow is releasing energy. And the energy that's released in catabolic reaction actually is stored in the form of ATP. So then later on, when your body wants to make stuff through anabolic reaction, the ATP then can be used for cellular work, okay? And then once ATP is used, it becomes ADP again, and then the catabolic reaction can turn the ADP into ATP again. So can you see how ATP, I have a next slide ready, it's kind of like a temporary storage for energy for catabolic reaction. So in case that you have more of the catabolic reaction going on than anabolic reaction, it'll be okay because it's like battery. Here's an example of battery, right? Catabolic reaction release energy, so it turn ADP into ATP. So in a sense, this scenario here, in catabolic reaction, you're charging a battery to full, okay? So then later on, when the anabolic reaction need to use the energy, they just pick up this battery, which is ATP, and then use it up and then become ADP again, and then catabolic reaction will charge it again. So this is a very good way to store energy when you don't need it, because when you need it, it'll be ready, right? So ATP serves as a temporary energy storage to allow the coupling of anabolic and catabolic reaction. So that's what we call energy. So that's how we talk about energy coupling and ATP cycle. ATP cycle allow the energy coupling between anabolic and catabolic reaction. Okay. So let's talk about cellular work. So what is cellular work? Well, it's work, right? So your cells constantly do a ton of stuff. Remember, I showed you that picture about how constantly transporting cargoes from Golgi and making proteins and doing all sorts of stuff. So when ATP become ADP, I show you that there's some energy that's released and there's inorganic phosphate, right? This phosphate, you should have kind of remember a little bit. When we transfer a phosphate to other molecules, we call this phosphorylation. In signaling, when we talk about cell signal, that's how it works. This phosphate get passed along. But another better example is, you remember phosphate has a negative charge. So if you put a negative charge on a protein, you're going to change its structure. Now, the example you already learned is sodium-potassium pump. Remember, sodium-potassium pump require ATP. Once the ATP bind to it, it turns into ADP, and the energy is used to pump sodium against its concentration gradient. And then at the end, the phosphate that's left on the pump change the structure of the pump, then allow it to bind to the potassium outside of the cell and bring it back in. Do you remember that? So this is one example of cellular work where you have the hydrolysis of ATP to power active transport. Okay, let's look at this worksheet. My internet was out for a few seconds. Let's see, what are some of the questions? Let's talk about, I think most of you get this question wrong. Well, number one, let's do this question first. So an electron will loses potential energy when it how would electron lose its potential energy? How would it go? Let's see. What about it has to do with the way it travels between the different electronegativity elements. Okay, so you need to think along that way. So an electron will lose potential energy when it moves to a less or more electronegative element. Right? Do you guys know which one? It's not going to be C or D, either A or B. Which one is it? So Aneri say that electron will lose potential energy when it, yep, B is correct, when you move to a more electronegative. Because remember, the more electronegative the element is, it holds the electron tighter, so it doesn't fly around as freely, so it has less potential energy, okay? So I think you guys are good with all except this one. For some reason, you guys can't find this one. If you look at PowerPoint, the answer for this one is right here, right? It's called phosphorylation. Or you can say it's said to be phosphorylated. That'll work too. Okay, so let's see if you guys have questions. We're done already, but I want to say a few things, so don't leave yet. I already talked about exam 5. It'll be ready by the end of the day, so you should get it done before next Monday. Okay? Make sure you study for it. Like last time, most of you didn't do very well. If you look up every single answer, you're not going to have enough time. Like I said, you still have to take your notes, organize your material. Same thing is open book, open notes. You cannot ask for help either through internet or your friends or other people. Okay. Today we're gonna have a study session, just like every Monday. We're gonna start at 11:30 using the same link that I have here. Let's see, log in. You're using the same study session WebEx link at 11:30, okay? Um, I want to know who will join the study session so then I know who to expect. Can you guys text me real quick here through the live chat so I know who will be showing up for the WebEx? And so next time on Wednesday, we'll talk about biofuels. So you guys need to have the biofuel soft chalk done. Um, is there any question? If no, you can go if you have no questions. If you have questions, stay along. Work on your worksheet, ask me questions so you can have it done. Okay, let me look at what I have here. Let me see I get it right. For 27, we're on week 12. Isn't this the 17? Yep, Ian, you're right. Let's fix this. There. So now I change that to the 17, which is Friday. I believe. See, I have this correct. Let's change that to 22nd. I think now when you guys do the biofuels, make sure you go to the biofuels project at the end here. And then the soft chalk, the lecture PowerPoint is here. The lab menu is here. Soft chalk is still 15, which is correct. So everything is correct now. Sure, Misty. Let's look at, are you talking about, let's see where it is. Let me open up that worksheet. So it's genetic number 2, right? So cellular respiration. Let me let me make sure I'm looking at right. Yes. Yeah, I got it right. So when is this one due? The 22nd. Yep, and you're right. So actually cellular respiration. So today we talk about metabolism. So that's one kind of the beginning of cellular respiration. And then we have the two main parts that talk about the different steps in cellular respiration. So yeah, so on the 22nd, that is the last class on cellular respiration. Okay, let me pull up that worksheet for you, Misty. So let me open it up soon enough. My computer is a little slow this morning. Don't know why it's not rolling. Why are you angry? If I can only pull up that worksheet. Oh, what if I go to the internet for help? Okay. Was it week? Week what? Here. Week. Was it week 11? No. No, not the right one. Must be 14. Okay. Wait, pedigree. Oh, here. Open it up. Okay, Misty, tell me whether this is the worksheet you're talking about. Oh. And you're wondering about this pedigree. Wait, where is it? Is this the one you're talking about, number 8, with the Huntington disease? Number 5. Oh, this one. Okay, this one actually is very easy. So if you look at this picture, last time you already know it is a autosomal dominant, right? Because the parents, one of the parents get it, and then most of the kids, 3 of the 5 kids will get it. So you know it's autosomal dominant. Now, so for autosomal dominant, you remember that to show the disease, all you need is one dominant allele. So this parent here could be homozygous dominant or heterozygous dominant, correct? Then for recessive, all you need, in order to be a normal individual, you need to be homozygous recessive. So this parent here has to be 2 small letter. Okay, so if you look at what number 5 is asking you, 1, 1, this picture doesn't show up right. This is 2, this must be 1. Why it looks like this? Picture looks all messed up. Can you not move this? Crop, no, red text. Oh, here. Okay, so if you look at this 1, 1, I guess it's talking about this guy, right? he has the disease and he cannot be homozygous dominant. If he is, then all of the kids will have the disease because all the kids will get one allele from this person. So then 1 will have to be, and the clue, so 1 has to be, I already tell you here, see, if individual 1 is heterozygous, right? So that's why if it's heterozygous, if it's showing disease, that means it has to be autosomal dominant. That's why it's A, okay? So I already tell you number 1 is all heterozygous, so it has to be big A, little a, right? Only need one dominant allele to show the disease. 1, 2, this person, the wife is normal. So like I said, normal person has to be homozygous recessive. If any person get one of these big allele, they will be showing the disease. So any normal people will be 2 small letter, right? Agree? 2, 3, 2, 3 here. This person is normal. So what would that person's genotype be? When just remember, Misty, when I ask for genotype in genetic question, as I mentioned in the live stream in the first genetic lecture, it's always 2 alphabets, uppercase or lowercase, right? Each alphabet represents one allele from the homologous chromosome. But if I ask for a phenotype, it'll be an adjective, a descriptive word: red, yellow, tall, short, got hair, no hair, extra thumb, no extra thumb, you know what I mean? So when in genetic questions asked for genotype, it's always 2 alphabets. Yeah, you got it. So for normal, this person, 2, 3, will be 2 small a, right? What about 3, 7? Let's look at 3, 7. This is easy. 3, 7 is also normal. So what will he has to be? Right on, you're getting the hang of it, right? Every normal person will be small a. Just remember in the case of autosomal dominant, which when I ask questions like this, if I say that if it's a heterozygous and it's showing the disease like this guy here, then it has to be autosomal dominant, right? Otherwise, it will be if we were autosomal recessive, the heterozygous will be a carrier and it will not show the disease, any symptoms. It will just look normal, right? He will be just a carrier. But if a heterozygous show the disease, so it has to be autosomal dominant. Is there another question on this worksheet? Okay, Misty, do we have more questions about this worksheet or are you good? Are you okay to do the to do question number 8. Now this is another question about autosomal dominant, right? Remember, which it just means that all it takes is one dominant allele for the individual to show the disease. It doesn't need two, it only need one. Okay, I'm going to end the live stream and I'll open up the WebEx in a minute at 11:30. Nobody said they're going to come, so I'm just going to open it up and see who will show up.